In this class, we will discuss about the anatomy of dicot root and monocot root. So, when you take the outer layer of the dicot root is made up of closely arranged parenchymatous cells, and this layer is known as epiblema. So, we have seen this outer layer is epidermis in the previous portion. In the root, it is known by a name called epiblema, and here and there some cells will give out some extensions, and these extensions are unicellular and they are known as root hairs. It will provide additional absorption ability for additional absorption capacity to absorb water from the soil. So epiblema, these cells, some of the cells will give out some unicellular extensions, and these extensions are known as root hairs. So below this epiblema you have loosely arranged, the cells should not be joined actually, loosely arranged parenchymatous cells, and this layer is known as cortex. So what is the peculiarity of this is it is arranged loosely, and hence there will be some spaces between the cells, and these spaces are known as intercellular spaces. So these cells are arranged loosely with some intercellular spaces. So this cortex is lined by another layer of closely arranged barrel-shaped cells, and this layer is known as endodermis. The cortex is lined by a layer of closely arranged barrel-shaped cells, and this layer is known as endodermis. So when you take some cells in the endodermis, it will be thickened in the tangential and radial walls. So it is thickened. Some endodermal cells will be thickened in the tangential as well as radial walls, and these thickened endodermal cells are known as Casparian strips. So these thickened endodermal cells are known as Casparian strips. So what is this thin walled cells? So these are known as passage cells. Passage means it will allow something. So what it will allow? So root hair will absorb water, and the water will travel along the intercellular spaces, and it will enter the passage cells, and it will enter into the inner tissue. So these passage cells will allow the penetration of water, which is absorbed by the root hair. So next to the endodermis is a layer of parenchymatous cells, and this layer is known as pericycle. So this layer is known as pericycle. So next to this, xylem and phloem will occur in bundles. So the small xylem will be on the top, while in the center it will be big one. In the bottom it will be big one. So it will be arranged as bundles. So actually you have to draw like this, two layered, and phloem will occur as patches alongside xylem. So this is the xylem and this is phloem. In xylem you can see here it will be a big one, here it will be small one. So it has two names. The smaller xylem is known as protoxylem, and the bigger one is known as metaxylem, and phloem will occur as patches alongside xylem. So this phloem and xylem are connected by a tissue, that is a group of cells called conjunctive tissue. So what is the function of this conjunctive tissue? Here it's joining for xylem and phloem, and this conjunctive tissue will later develop into cambium or cambial cells. Cambial cells are last class we have seen it is capable of producing the secondary, it is capable of regaining its meristematic activity, and it is capable of producing new cells that will result in secondary growth. So what is the secondary growth in plant? So when you take a neem plant, small neem plant, its stem will be tender, but when you take a neem tree, it will be big, right? So it is, we will call that as a bark. So bark is actually a result of secondary growth, and this secondary growth is done by the cambial cells, which is formed from this conjunctive tissue. So this makes the dicot root capable of doing the secondary growth. So root also, you can see it will not be a tender one. The root of the neem plant, it will be a hard one, and it also has some outer brownish covering. So that is what is known as a secondary growth. So in here in the center you have a small pith. So here center you have a small pith. This is what is about dicot root. So outer layer is epiplemma. It has unicellular extension. Some cells have unicellular extensions called root hairs. Below this epiplemma are loosely arranged cells with intercellular spaces, and these cells are constitute the cortex. The cortex is lined by endodermis. These are made up of barrel shaped cells. Most of the cells of endodermis are thickened at the tangential and radial wall with the chemical. I didn't mention this. So it is thickened by a chemical called suberin, and these thickened cells of the endodermis are known as Casparian strips. So thin-walled cells of the endodermis are known as passage cells. So next to the endodermis is again closely arranged parenchymatous cells called pericycle. So below this comes the vascular bundles. So xylem and phloem occur as separate bundles here, which is joined by a special kind of tissue called conjunctive tissue. Conjunctive tissue will later develop into the cambial cells, which will regain its dividing capacity and show the secondary growth. The smaller xylem are known as protoxylem, and the bigger one are known as metaxylem. So in between in the center you have a small pith. So the cells or tissues below the endodermis, all the tissues, so pericycle, vascular bundles, pith, so all these below the endodermis are collectively known as stele. So all the tissues below endodermis are collectively known by a special term, stele. So this is what is about dicot root. So what about this monocot root? So let's have a differentiation here. You can see this is a dicot root and this is the anatomy of monocot root. The epiplemma, root hair, cortex are similar. Even endodermis, pericycle all are similar. So what is the main difference here? The number of xylem bundles. Here you can see only 4, but here it is 8. So usually the xylem bundles in a monocot root will be 6 or more than that, and it is known as polyarch xylem. So which is more bundles. Xylem will occur in more bundles when compared to the dicot root. And here the pith will be more. Pith is big and well developed. In the case of monocot, the pith is well developed and it is more in number. And another main difference is here we have seen a special tissue called conjunctive tissue, which will produce the cambial cells, but here the conjunctive tissue is absent and hence the secondary growth is not present. So this is the main difference. So when you take both the dicot and monocot root, the root, hair, epidermis, cortex, endodermis, epidermis is also known as epiplemma, so no issues. In the case of root only you will use the term epiplemma. The other word for epidermis in root is epiplemma. You should not use that in stem or leaf. Here the root, hair, epidermis, cortex, endodermis, pericycle of both dicot and monocot root are same. And what is the main difference? The number of xylem bundles. When compared to dicot root, monocot root will have more number of xylem bundles, and the pith in a monocot root is more when compared to it is large and well developed when compared to dicot root. And main difference is the absence of conjunctive tissue in monocot root, and hence the cambium is not produced, and hence there is no secondary growth in a monocot root.